Hello everyone, and welcome to the second part of creating new neurons in the adult brain. So, following the last question, the strategies to create new neurons are the following. Either we create neurons in cell culture, so artificially in the laboratory, and then we implant them in the brain, and this process is called neurotransportation, or we create new neurons inside the brain. And there is two techniques to do so: endogenous cell replacement or in situ reprogramming. Both of them will lead to the creation of new neurons inside the brain. Let's talk first about neurotransplantation. So, there's two ways of obtaining neurons grown in the lab, so artificially. There is, to give birth to neurons, you need first to have neural stem cells. Neural stem cells come either embryonic stem cells or induced pluripotent stem cells. Let's focus first on embryonic stem cells. So, embryonic stem cells actually are the result of a fecundation between a spermatozoid and an ovula. Then it will give birth to an egg cell, which afterwards will divide and form a morula. And then it will give birth to a blastocyst. Well, it will transform into a blastocyst, and inside the blastocyst you will have an inner cell mass, which will be composed of embryonic stem cells. These embryonic stem cells are what we call totipotent stem cells. Totipotent cells, means that they can actually replicate, but also give birth to any cells of your type. So you could give birth to neurons, for example, neural stem cells, muscle stem cells, a lot of different kinds of cells. And so those are really interesting because with these cells you can generate neural stem cells and then afterwards generate neurons. The problem with embryonic stem cells is that for a human, you would need a human embryo. And it's kind of difficult to come by a human embryo these days. Especially, there's also a very, very important thing to consider when using embryonic stem cells are ethical issues also. So it's difficult to obtain embryos because of practical considerations, but also because of ethical considerations. So, as an alternative, Shinya Yamanaka, which you see on the picture on the right, who won the Nobel Prize in 2012, thought, okay, embryonic stem cells are a bit difficult to come by. Why? What if actually we just use cells from patients? So we take some skin cells, we will make them grow, and we transform in a sort of state which looks like embryonic stem cells, and so it's called induced pluripotent stem cells with a cocktail of molecules. That way, we have no problem with ethics. We can actually generate as many cells as we want, and from the patients, and that is actually very interesting because you have less risks of rejection, or graft rejection, if you need to put them back to a patient, for example. So these induced pluripotent stem cells are very interesting because they can actually self-replicate and give birth to any cells you want, like embryonic stem cells. And here we can actually give birth to neural stem cells if we want to. Okay, so how does that work? Well, so you have your induced pluripotent stem cells, you transform them into neural stem cells, and after that you obtain predefined neuron, and it's very important to obtain a predefined neuron because there's multiple kinds of neuron in the brain, and you have to make sure that the one you're giving is the right one. And after that, once done, you transplant them into the brain. Okay, great. What are the applications? For example, here an interesting application is implantation of human neural stem cells in patient who had stroke. So patient who lost cells after a stroke. And it has been seen that this patient, when there were implanted human neural stem cells inside the brain, they had a recovery, a motor recovery. So that means that we could actually move back or have a better motor movement, which they didn't have before when they had a stroke. So this is really interesting. Another application is human neural stem cell transplantation in chronic cervical spinal injury. So spinal injury is one of the most one of the biggest application for neural stem cells. So spinal injury results in impaired motor movement. So people who had, for example, car accidents and their spinal are totally damaged, they lose the control of their legs or their arms sometimes, both in both cases. So it could be kind of very debilitating. So it has been shown that the transplantation of human neural stem cells in this patient could ameliorate their movements, their motor movements, and it's kind of very, very interesting and encouraging results. So both results were done in humans, and this is actually very promising research. I will give you a quick review, a quick summary of the advantages and the drawbacks of such technique. So the advantages is that there's expertise. There's been a lot of research that's been done on human as well, and this is very positive. This is really positive. So that means that there's a lot of data that we know, and for creating new treatments, it is very important to have the best understanding of what you're actually doing. Then it has been so, and as I illustrated before, then there is a restoration of functional phenotype. However, there is some drawbacks. So the grafted neuron needs to be of the right type, and it's sometimes difficult in vitro to be sure, am I giving the right neuron? So there needs to be more specific techniques to identify what neurons I have in front of me in the end. When you're transplanting grafts, so neurons inside the brain, it could be quite invasive, and so it could not be sometimes always be practical to do. Graft survival is also a challenge. We have saw that neural stem cells are generated in a culture medium, so in artificial conditions and not in vivo. And there's a huge there's a huge gap between the in vitro environment and the in vivo environment. And this gap is sometimes so big that it could actually impair the survival of a graft inside the brain. So this is also a technical challenge. Finally, there is sometimes a risk of tumor formation using induced pluripotent stem cells. As I mentioned before, induced pluripotent stem cells can replicate themselves, and cells which are derived from this induced pluripotent stem cells can do as well. The problem is it could actually replicate and multiply themselves without control, and cells which multiply themselves without control can form tumors and then cancer. So it is very important to make sure that our cells are not going to do so, and this is not entirely sure yet. Another alternative, so following this approach, is to create new neurons inside the brain. Okay, so how does that work? So people thought, okay, generating the neurons in vitro and then grafting them in vivo is nice, but in the brain, there's already neurogenesis going on. So why not just simply recruiting neurons from neurogenesis sites? Well, how does that work? So you increase neurogenesis in the submatricular zone and the subgranule zone. Okay. However, when you do so, you have a risk that the generated neurons are not always matching with lost neurons. Indeed, the neurons which are generated in these sites are very specific, and they're not always matching with other neurons of different areas. So this is an issue. Another issue is, okay, I have generated a lot of new neurons, let's say. Okay, how am I going to be sure that my neurons are going to migrate to this place, for example, in a live brain? So this is also a technical challenge as well. So following this approach, some scientists thought, okay, why not just creating neurons by converting glial cells into neurons inside the brain? Why glial cells? Well, glial cells, I can see in this picture, are everywhere in the brain. So they are here, 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 and here. So that means that if I have lost some neurons, I don't know, here, well, all I got to do is transform glial cells precisely here, and I have new neurons there. Simple. How do I do that? Well, you use a viral vector here, okay? And this viral vector is going to contain a transcription factor. And this transcription factor with this viral vector is going to be injected in the brain and is going to infect my glial cell, and this glial cell is going to express the DNA of my virus, or the transcription factor, and this transcription factor is going to differentiate my glial cell into a neuron, and so it's going to convert it, basically. So that's great. But what glial cell should I target? Well, actually, a very interesting candidate are astrocytes. If you remember, astrocytes have many roles in the brain, but as I mentioned in the last video, astrocytes also can create new neurons, and they can do that in the sites where neurogenesis happens in adults, which is the subventricular zone and the subgranular zone. So this is interesting because that means they will share common features with neurons. And so another thing which is really nice from these cells is that there are prevalent cells in the brain. So there's many astrocytes. So that means that potentially there's many astrocytes which could be converted to neurons if needed. So that makes them an interesting candidate to be converted to. Another candidate is oligodendrocyte progenitors. So oligodendrocyte progenitors are cells which can give birth to oligodendrocytes. And the advantage with oligodendrocyte progenitors is that they can self-replicate. So they can self-renew for basically a very, very important numbers of times. So that means that if you convert one oligodendrocyte progenitor, you could generate multiple neurons. And that is really interesting because the more neurons you generate, the happier you are, because the bigger the chances you have to recover from a lost function. And a plus there is that it shares common ancestry with neurons as well. So it's really easy to convert them into neurons. The problem there is that this self-renewal potential decreases with age. So it may be not the most appropriate candidate when you are doing this in older patients. So how do we convert glial cells into neurons? So let's take a closer look. You have glial cells there on the left. Okay. Then you have your viral vector containing your neurostem transcription factor. However, depending on the transcription factor you use, the process of cell conversion is not going to be the same. Indeed. For example, your virus contains NeuroD1 and infects your glial cell with NeuroD1. What happens is that your astrocyte, for example, is going to be transdifferentiated. That means that it's going to be directly converted to a neuron. So one glial cell, one neuron. The problem with such technique is that you will need to convert a lot of glial cells to obtain a lot of neurons. And the problem is, let's say that you have 100 glial cells. And you have 100 glial cells. Well, if you convert 100 glial cells and you generate 100 neurons, who will take care of your neurons? Because they will be alone. There's not going to be any more astrocytes to take care of them. And astrocytes are crucial for the current function of neurons. So this is the problem with such technique. So instead, if you use another transcription factor, which is called SOX2, well, you could transform your cell into an active progenitor state. That means that the glial cell will not directly be converted to a neuron, but to a neurostem cell. And this neurostem cell will be able to self-replicate and generate multiple neurons. And this is really interesting because with one glial cell, you can obtain a multiple number of neurons. And so you have no risk of creating an imbalance between glial cells and neurons. Let's have an example there on this in situ conversion of neurons by looking at Huntington's disease. So, Huntington's disease is a disease where you have a loss, so a neurodegeneration of neurons in the cortex and the striatum. We're going to focus on the striatum there. So the striatum is a place which is involved in the control of motor movement. There's two kinds of cells there. There's activating cells and inhibiting cells. When you're making a movement, your movement relies on the inhibition or activation of your movement. And it relies so on activating cells or inhibiting cells. The thing is, Huntington's disease, you lose inhibition neurons. That means that when people who have Huntington's disease, they make uncontrollable, sometimes very violent movements, which can be very, very, very difficult to deal with in everyday life. So they have impaired motor movements, and this concerns between 4 to 15 patients per 100,000 individuals in average. And so scientists thought, okay, what if we try to restore this inhibitory neurons in the striatum and see if we could actually restore the phenotype and the function of a normal, healthy brain. So here's an article. Research was done in 2020, and it's a gene therapy, so a virus containing a transcription factor which will convert striatal astrocytes into inhibitory neurons, which are GABAergic neurons, in mouse models which have Huntington's disease. Let's see what were results. So they saw in this paper that there was an improvement of motor function and survival rate in Huntington mice. On the figure G, so in this picture there, wild types means normal mice, so mice which don't have a disease. You can see that this square here with a lot of lines there, this actually represents the movement of mice in a closed space. And you can see that there is a lot of movement. Basically, it's like the whole square is practically red. But the other mice, which have Huntington's disease here and there, you can see that they're not moving so much, actually. Their movements are really much reduced. In the mice which receive a treatment there, so the gene therapy, you can see that there is a significant improvement of motor movements compared to the Huntington's mice. And this is really, really interesting. Other data showing more or less the same results. This is a bar plot showing the travel distance centimeters and in average in mice which receive a therapy. And so this is are the wild types, and these are the mice which have Huntington, and these are the mice who received who have Huntington and receive a therapy. And you can see that the travel distance is doubled compared to mice which did not receive a therapy. So very interesting results. And here on the right, these are the mice. So 7 days before surgery, so 7 days before they receive the treatment. So this is the body weight, and you can see that more or less the same body weight. But 30 days after surgery, the mice who receive a treatment have an increased body weight. What does that mean? That means basically the muscle mass has increased. And if muscle mass has increased, that means that the motor functions in the brain are partially or at least ameliorated. So this is interesting data as well. And finally, I would like to conclude on the fact that the survival in these mice which receive a therapy is also increased. They have much bigger survival rate chances at 90 days, which is not the case for mice which now receive a therapy. So those are really encouraging results, and it would be really nice if this could actually be proven, it could be actually repeated and then proven in humans. I will give you also a summary for the in situ in vivo programming. So the advantages of such a technique is that it's less invasive than cell transplantation. It could potentially convert the right neuron at the right place. into the right neuron at the right place, it could ameliorate the functional phenotype. However, there are some drawbacks. There's much more research that needs to be done. There's been no clinical studies which has been done in primates so far, and no clinical studies in humans. So a lot of data needs to be acquired to fully understand the whole extent of such therapy. There's also needs to be some technical improvements which needs to be made to confirm generated neuron origin. Indeed, some papers and some articles have casted doubt that the neurons they generated, so they converted, are not actually so many as they think. They think that some neurons they actually created were already there. What happens is that it is not entirely sure if the neurons which were saw after the therapy, for example, were neurons which were already there, or if it was neurons which were converted from glial cells. So there still needs to be some technical improvements to confirm that. Another thing is that the conversion rates needs to be improved as well. Indeed, the conversion rates to convert glial cells into neurons are quite low, and there's a lot of barriers to overcome these issues, which needs to be overcome to increase these conversion rates. And it's possible, but yet it's not entirely done yet. So it still works to be done in this path. And finally, generated neurons. Can maybe be detrimental to pre-existing neurons. It is not sure that if you generate new neurons, these neurons are going to integrate properly and are going to form the right synapses with right neurons. It could be possible that they could actually form wrong synapses with other neurons, and it could actually impair pre-existing circuits or pre-existing functions. So there's still some work which needs to be done to confirm that this is not really happening. So I would like to conclude on this presentation. Both technique, neurotransportation and in situ reprogramming, are very promising techniques, and potentially they could help and maybe cure some diseases where it was not possible before, specifically, for example, in traumatic brain injury or in neurodegenerative diseases. However, there still needs to be a lot of work which needs to be put in place to make sure that these therapy are the safest and the most efficient as possible to be used in humans. I would like to show you a quick video that I found on YouTube, which actually shows you how much the network formation of neurons is complex and beautiful at the same time. So you can see here that axons and dendrites sprouting from neurons and trying to find their way to communicate. And it is beautiful and so complex. When thinking about it, you can actually think it's a bit like also the network of a city in the end. So you can really see that first of at the beginning of the video, it's more or less disorganized. It goes a bit everywhere, and then it kind of like starts to focus at certain points. You can see that it's more or less like there's like some main, some main roads, so to speak. Okay, so if you want to go further in details in following this presentation, I have selected two papers which are kind of interesting if you want to have more more opinions or more knowledge on in situ conversion of glial cells into neurons. So this paper actually is kind of interesting because it's the one who illustrates the doubts that we have on the origin of converted cells and what would be potentially maybe the solutions to overcome such issues. And this is another article showing another application of in situ conversion of astrocytes into neuron in another neurodegenerative disease, which is Parkinson. Thank you for your attention. I hope you enjoyed the video, and please feel free to leave any comment, suggestion, feedback, anything you want. It's much welcome. It's very welcome actually. Sorry. Because it's my first video, so any feedback, anything, it's really welcome so I can improve and do better. I hope it was of use, and I hope you learned some few things, and I hope it gave you some new neurons as well. Here are my sources. So just a really nice fountain there, so hope you appreciate it. No, just kidding. Here are my sources, so feel free to dig in as well if you have more, if you want to know more. Thank you, and hopefully see you at the next video.